Hi everyone. I hope I'm audible. Let me just check here. Yep. Alright, so let's get started. So let me introduce myself. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. So join with us in Telegram. So that is for homework and class notes. You can join the channel Vani Kari ICSE class 9 and 10 biology and the Telegram channel of Unacademy ICSE and ISC both. So this link you can find in the description box. So go to the description box. You can find the link for Telegram channel. You can find the link for special class, community as well as Bucks Bounty. So you can find link of all these and join the respective channel. So Bucks Bounty also, if there is any inappropriate content, so you can go ahead with that as well. So special classes, you can basically learn from your favorite educator for free. So basically these classes are free classes where you can learn for free from different educators, as different educators will come up with a different theme based sessions. So you can engage with the educators via poll. So you can also clear your doubt in the live special class. So here for this separate question tab is available. So you can click on that question tab. You have an option to type in your question, attach an image and send it to the educator during a live session, so that you can clear your doubts during the live session itself. And now for the top special classes, you can visit the link in the description. So this is about the Unacademy subscription feature. One second. Yeah. Yeah. So where you can learn live from the comfort of your home. Unlimited access to all the courses will be there. Then top educators of India are going to take the classes in one platform. So that is Unacademy platform. Regular doubt clearing will be done. That is after about every 3 sessions. The fourth session will be a doubt clearing session. So whatever doubts you have related to the topics that are previously covered, so you can clear your doubt in these plus classes. That is mainly a doubt clearing session. So complete syllabus coverage will be done. Okay, so here it is 100% syllabus coverage will be done, whereas in this one only a certain percentage. Okay, so only a certain percentage of topics are covered under the special classes. So all you can go is for the plus classes, where 100% syllabus complete can be done. So mentorship and guidance can be given. Study material, that is in the form of PDF, will be uploading, so you can download that PDF. And also the practice tests can be given. Live test series are given, and also the patch courses. So here I'm sorry. So we have a batch in English, we have a batch in English. So you can go for any of these batches, so depending on your preference. So there is a daily practice section, weekly mock test series is also conducted, live quizzes, daily MCQ and subjective test series are also there. And we have an offer right now. So this is ICSE combo offer, so you can start your preparation early there. So now for class 9 you can go with the 1 year subscription. Okay, and also you can start your preparation early for JEE as well as NEET and NTSC. So this is again for this also you can take a 1 year subscription. So both of them together it is where you will be going for class 9 classes, along with that the NEET JEE as well, where the cost is basically 34,000, which is being crossed. And since the offer is there, so you will be getting it at 31,050, and with a code, so that is Vani YT. So you, okay, so 1 second, so it is Vani YT. So there is a offer that is going on, so you can make a best use of this offer. And also we have an offer where you can get 20% discount on the subscription. So plus subscription, this offer is gonna end on 30th of June. Okay, so if you are going for 12 months of subscription, so basically it is not 17,250, it is 13,800, where you will be saving up to 3,450. That is with the referral code Vani YT. So those who are planning on taking a subscription, so you can go ahead with this. So using this code you will be getting 20%, and this is the amount that you would be paying for 12 months. Okay, so if you just want to go for 9 months, so there is this option also. For 9 months it is basically 11,960, and you will be saving up to 2,990. So you can go ahead with this as well with the referral code Vani YT. So those who are planning for class 9 as well as for class 10, so maybe you can go with the higher months of subscription here. Okay, so this is the regular pricing, and this is my code Vani YT to get 10% discount. So today we'll be discussing about the cell organelles. Okay, that is the cell organelles, so that is especially in a eukaryotic cell. Okay, so this is about the eukaryotic cell. So in eukaryotic cell you can find a well defined nucleus. So here this one is a nucleus. Now this is a cytoplasm, this is a cell membrane. So what cell is this? So this is an animal cell because there is no cell wall here. So cell membrane, nucleus and the cytoplasm, which is basically a protoplasm itself. Okay, so this is a protoplasm. So now main features of a cell when we mention, so these are all of this together, so we call this to be a protoplasm, which is a living content of the cell. So this is a living content of the cell, and suspended in the cytoplasm you can find many organelles. So that is a cell organelle. So cell is composed of several membrane bound structures. So what are those membrane bound structures? Anybody? So what all membrane bound structures you can find in the cytoplasm? I'm really sorry. I'm having a cold and cough and also a fever, and that's the reason I'm a little on lower side. Okay, so membrane bound organelles when we mention, so you can see there is a cell. Okay, so this is the cell. Let's say there is a nucleus here. So in continuity with the nucleus you can find endoplasmic reticulum there. So this is going to be a endoplasmic reticulum. Okay, hi Prince, hi Pratham, good evening. Okay, so this one be the endoplasmic reticulum. So arising from the endoplasmic reticulum itself, you can see there is a Golgi apparatus formation. So this is arising from where? The Golgi apparatus is basically arising from the endoplasmic reticulum. So in that case, you can find a number of stacks here. So this is a Golgi apparatus having its own vesicles there. Now arising from these, basically you can see there is a formation of lysosome, there is a formation of the vacuole also. So since we have drawn an animal cell here, so there can be many small vacuoles can be present, or they can be totally absent. So this one, let's say this is a lysosome now. Okay, so this is lysosome is coming from where? Lysosome is basically formed from the Golgi apparatus. So this, all this we consider under the endomembrane system. So why do we call this is an endomembrane system? That is because arising from this particular outer membrane of the nucleus, attached to that itself, you can find there is the endoplasmic reticulum. So let's just discuss about that. And before that, let's have a look at the eukaryotic cell. So size, it is gonna vary between 5 to 100 micrometer. Okay, so nucleus is well defined. So why do we say that it is well defined? Because there is a outer membrane. So this one is the outer membrane, and then there is the inner membrane. So you can also find there is a inner membrane. Okay, and both of them are fused here. So there is a fusion between the outer as well as the inner membrane. So there is a nuclear pore. This one is basically a nuclear pore. Hi Lakshman, good evening. All right, so nuclear pore is also there. So now with respect to all of this, you can see there is basically a nucleus is there. Okay, so this is a nucleolus, this is all nucleus, and here inside this, I'll just highlight this just a second. Yeah, so this whole part what you see now, so this is basically a nucleoplasm which is found inside the nucleus. So what is this tiny bit then? So this is a nucleolus. So now with respect to that, this is a well defined nucleus, where you can see that there is a DNA suspended inside this nucleoplasm. So number of chromosomes, so this is gonna vary, but S anyway it will be more than one in most of the living organism. The type of a cell when we mention, so this is an advanced one. So why is this an advanced cell? So why do we say that this is advanced? Because you can see different organelles. So it is having a well defined nucleus, it is having an endomembrane system. Now number of cells when we mention, so there can be single cell or there can be many cells as well. Okay, so there is a single cell, there is many cell. You can find, it's not that eukaryotic cell when we mention, so it's not that basically there has to be a single cell only. So you can find eukaryotic unicellular. Okay, and also you can find eukaryotic multicellular organisms. So under, so this is multicellular. Under unicellular you can find there is basically the protistas, like amoeba is there, paramecium is there, and you can also find the euglena. So different organisms which organisms which are unicellular but they are eukaryotic. That means they have this well defined nucleus. Now multicellular when we mention, so multicellular organisms, many of them. So starting from the fungi, then there is a plantae kingdom, all the organism, then you can find the animal kingdom. So basically animalia, plantae, fungi, so all these are multicellular and they are eukaryotic. That means they have a well defined nucleus. Okay, so now the number of cells, so there can be single or many. Membrane bound organelles are there, so that is present. So that is basically the endoplasmic reticulum, Golgi body, then lysosome, then there is a vacuole. So all these can be considered under the membrane bound organelle. So now ribosomes when we mention, so they can be larger ribosomes, you can find are attached or free. So let's say here is the cytoplasm now. So this is the cell. Okay, and inside this cell, so basically this is full of cytoplasm itself, right? So this will be having what? This will be having a cytoplasm, because the outline that I have drawn, so that is basically a cell membrane or a plasma membrane. So inside this completely you can find a cytoplasm there. So this is gonna be a cytoplasm. And now when we mention about the cytoplasm, so let me just cover this up. So you can find the ribosomes to be free or they can even be attached. So how exactly that happens then? Like let's see where exactly synthesis of ribosome occurs. Anybody has any idea on this? Where the synthesis of ribosome occurs? No idea? So now here you can find a nucleus. So this one, let's say this is the outer membrane of the nucleus, and there you can find the inner membrane of the nucleus, which is a nuclear envelope. So we call this to be a nuclear envelope. Now who synthesizes these ribosomes? So ribosomes are being synthesized, so that is by the nucleolus. Okay, so nucleolus is the one that synthesizes a ribosome. So here the synthesis of ribosome occurs, and those synthesized ribosomes, you can see that they will start getting, like they'll attach onto the surface of nucleus as well. Okay, so we can just draw few ribosomes over here, because they'll get attached even onto the surface of the nucleus. And the ribosomes which are synthesized here in the nucleolus, so they'll be passed through this nuclear pore, and throughout this cytoplasm you can see there are many ribosomes which will be found here. Okay, so there can be many ribosomes which are found throughout this, which is basically again synthesized by the nucleolus. Now in continuity with the outer membrane of the nuclear envelope, so you can find there a structure, so that is basically a membrane like structure. Okay, so that membrane like structure is basically this one, so which is a endoplasmic reticulum. So why is there a name called as endoplasmic reticulum? Any idea on this? Why is it named as a endoplasmic reticulum? No idea? So endoplasmic reticulum, endo is what? Endo meaning is, I'll just write it here. So endo is basically inside. Okay, and plasmic is what? Inside the plasma. So endoplasmic, when we mention, so this is inside the plasma, you can find a reticulum like structure. So what is that reticulum like structure? So reticulum is nothing but a network like structure, and hence the name is given as the endoplasmic reticulum. Okay, so that is inside the plasma, so there is a network, and this network is basically a endoplasmic reticulum. So now you can find attached ribosomes onto the surface of this endoplasmic reticulum. So let's say here the ribosomes are attached randomly. Okay, I hope you can see the color difference over here. Thank you, Suyog. So this is basically a endoplasmic reticulum which is attached onto the surface. I think this can be seen, right? Or maybe I can just change the color. Just a second. Yeah. So here the ribosomes are attached onto the surface of the membrane, and now when we observe it under the microscope, so this gives a rough appearance. Okay, and hence this is called as a rough endoplasmic reticulum. Okay, now on this side, so let's say there is no attachment of any ribosome, so this side can be said as a smooth endoplasmic reticulum, because again when we observe it under a microscope, this appears to be smooth. And this is anyway a nucleus. So now in continuity with this particular endoplasmic reticulum, so there is a formation of Golgi body. So Golgi body formation is basically from this itself, which is arising from the endoplasmic reticulum. So now this is the Golgi body which is formed here. So all these are ribosomes. I'll just label this. So this one is anyway ribosome. Maybe I can just change the color of the ribosome to red itself everywhere, because here I have represented it with red. Okay, so that you don't get confused. So this is everything over here is basically a ribosome. Okay, so ribosomes are there suspended in the cytoplasm. So they are also attached onto the membrane of the nucleus. They are also attached onto the membrane of the endoplasmic reticulum. Alright. So now hi Prince. Good evening. Okay, so now this endoplasmic reticulum is giving rise to a structure called as a Golgi body. So now once the Golgi body is formed, so now whatever protein is synthesized. So let's say here, Mm, this is a ribosome now, where the function of ribosome is what? Function of ribosome is protein synthesis. So protein is being synthesized by the ribosome, and now for that reason, as the protein is being synthesized here, so this synthesized protein is getting modified here in the endoplasmic reticulum. So modification of protein you can see towards the rough endoplasmic reticulum. So let's just write this modification of proteins. Okay, so modification of protein is done where? So that is in the rough endoplasmic reticulum. And now if we talk about the smooth endoplasmic reticulum, so this is responsible for lipid synthesis. Okay, and the modification is also done with that. And as the modification occurs, so this modified protein is now gonna enter the Golgi body, and there is a packaging of this particular structure. So that is basically modified proteins are packed here by the Golgi body vesicle. So vesicles are there which is from the Golgi body itself. So this is a Golgi apparatus or Golgi apparatus. So in that case, modified protein will be stored in here, and these are basically can give rise to a vacuole. So let's say we'll draw a vacuole here. Okay. So this is basically vacuole formation you can see. Then there is lysosome formation you can see. So lysosome is being formed. So this is basically arising from the Golgi apparatus itself. So in that case, let's label this to be a lysosome, and lysosome is basically hydrolytic enzyme. So this will have many of the enzymes. So what exactly is enzyme? It is a modified protein. So enzymes are protein in nature, and those enzymes are packed here. So function of Golgi apparatus, we can say packing of these modified protein into vesicles, so which will be basically lysosome. Or if we are talking about a plant cell, so there can be vacuole formation, or in animal cell also there can be vacuole that is present, but these vacuoles are small and they can even be absent as well. So this is in case of an animal cell. So the one that I have drawn here is an animal cell. Okay, so this is all comes under, so like nucleus is there, endoplasmic reticulum is there, Golgi body is there, then there is lysosome, you can also find there is a vacuole. So all these are considered to be under the endomembrane system. So why do we call this to be a endomembrane system? Each one of them are giving rise to each one of them, like now endoplasmic reticulum give rise to a Golgi body. So Golgi body gives rise to a lysosome, and also this one that is basically a vacuole. So these are placed under the endomembrane system. So now separately other than this you can also find, so this is basically a cytoplasm itself. So I'll just label. Now inside this you can also find there a mitochondria is present. So mitochondria is again a double membrane structure. Okay, because it is having a outer membrane, and the inner membrane is basically folded to form a cristae like this. Okay, so inner membrane is folded, so there is a inner membrane which is forming the crista or a cristae. Okay, and this one is the outer membrane, and on the whole, so this is basically a mitochondria. Okay, so mitochondria is, what is the function of mitochondria? Anybody? Yes, so lysosomes are also called as suicide bags because let's say any germs that enter our body. Okay, so these lysosomes, since they are having a hydrolytic enzyme, so they combine with this particular pathogen which has entered. So they'll combine with this, and the digestion of this particular bacteria is done. So it can be bacteria, it can be fungi, it can be virus, any foreign object. It even responds to the dust particle as well. And also This one I said only in terms of infection. But if there is a old cell that is present, or if there is any old mitochondria, let's say organelle when we mention, so if a old mitochondria is there, so this particular lysosome will go combine with the mitochondria, and it will kill this mitochondria, because this mitochondria might not be useful at all, because that is not doing its function. This is lost, like it has lost its capacity to perform the function. So in that case, lysosome is going to digest this mitochondria. Lysosome can also digest the foreign particles entered. Lysosome is also going to burst open and kill the whole cell, and that's the reason we call this lysosome to be a suicide valve. Yes, so this is basically mitochondria. We call this to be a powerhouse. So why exactly do we call this to be a powerhouse? So if you have studied respiration chapter already, so in respiration chapter what is mentioned? So there is a glycolysis process that occurs in the cytoplasm, right? So in the cytoplasm you can see there is a glycolysis process occurring, and the Krebs cycle. Okay, so this is about the Krebs cycle. So Krebs cycle occurs when? So that occurs in the mitochondria. And since the mitochondria is the place of the Krebs cycle, the result of Krebs cycle is basically large amount of ATP production. So that is for 1 mole of glucose. Okay, so there is 38 ATP are produced. So there are so many ATPs that are produced here itself in the mitochondria. So that is inner membrane of the mitochondria. So let me just explain it here. Okay, so now this is the cell, and here is the mitochondria over here, so which is the inner membrane is folded. So in the cytoplasm glycolysis occurs, where the glucose is converted into pyruvate. Now this pyruvate which is synthesized here, so this will go to the mitochondrial matrix that is inside, and here you can see the Krebs cycle occurring. So Krebs cycle is the one that synthesizes a large amount of ATP, and that's the reason we call this mitochondria powerhouse of the cell, because a huge amount of, that is basically 38 ATP are produced just from 1 mole of a glucose. Okay, so large amount of ATP are produced by the mitochondria, which is going to supply the ATP to the rest of the metabolic process that occurs in the plant body, animal body, or any living organism. Okay, so in that case ATP is a must for any metabolic process to occur. Yes, ATP is adenosine triphosphate. Yes, that's right. Okay. Now, other than mitochondria, you can also find there. So there is a presence of plastids. So this you can find in a plant cell. So let's say here itself I'll draw and explain. So now plastids, when we mention, that is a chloroplast. So there is a cell wall, okay, and then you can find a cell membrane is present. So cell wall, cell membrane is present. I'll just draw the chloroplast. So rest of the things are anyway gonna be same. Lysosome might not be there, but the chloroplast. So now I'm trying to explain you the chloroplast structure. So that is basically a plastid type, that is chloroplast. So in that case you can find a double membrane structure here again. So this is basically a double membrane structure where you can find a outer membrane, then there is a inner membrane that can be found. So these two together, so these are forming a double membrane of the chloroplast. So this is basically chloroplast, which will have different substances, like the ground substance, just like cytoplasm, which you can see in a cell. So here the ground substance will be there. So that ground substance is called as a stroma. Okay, so this one is a stroma is found here. So this one I'll just label this part, just a second. So this is stroma, and there are chlorophyll. So chloroplast contains chlorophyll, right? Any idea where exactly you find chlorophyll in the cell, that is in the chloroplast? Any idea about this, where exactly chlorophyll will be present? Excuse my cold. Anyway, So I'll just explain, if you, even if you don't know, that's not a problem. So now you know there are structures called as thylakoid. So many stack of thylakoid, it is called as a granum. Okay, so you can find a granum there, and these particular whole thing, so now this whole thing is called as a granum. Okay, so this one is a granum, and inside this granum itself, that is in the thylakoid wall, so this is a wall of a thylakoid, so there itself you can find the chlorophyll that is present. So chlorophyll is present in the chloroplast, and this is the one which is going to trap the sunlight. Like you know, the chlorophyll traps the sunlight and excitement of an electron you can see, and that excited electron is going to pass on the energy for the photosynthesis process. So now with respect to that, this is now called as a granum, where the individual unit, so this is now an individual unit, so individual units are called as a thylakoid, and thylakoid is where you can find the chlorophyll. Okay, now you know everywhere, so it's not only here the thylakoid, the chlorophyll is present. So this chlorophyll is present here everywhere, like you know, all the structures over here, that is all the thylakoid over here will have that chlorophyll, and there is a excitation or excitement of the electron that can be seen. So like this, there are many of the grana. This is a granum. So now you know they are connected to each other, so that is by the fret or the stromal lamellae also we can call. So this is the structure of the chloroplast. So chloroplast is there, comes under the plastid, and also there is a chromoplast. Okay, so there is chromoplast, there is chloroplast, and there is a leucoplast. Anybody knows what is chromoplast or what is leucoplast? No idea on this. So chromoplast is not the green pigment containing one. So this is, you can find colored pigments. Okay, so colored pigments can be there. So those color pigments are nothing but, there can be carotenoid. Okay, so there can be carotenoids can be there, or you can also find xanthophylls. So different pigments can be found in this. Just a second, I'll drink some water. Yes. So chromoplast departs color to the floral parts. So you see basically the petals are colorful, right? So there is a red color petal, yellow color you can find, purple color you can find. So there is a anthocyanin pigment also. So depending on the pigment, so there is a coloration of these petals or the flower coloration. So this is gonna change. So these are basically colored pigment you can find in a chromoplast. Chloroplast, it is basically a green pigment, and leucoplast, when we mention, so this is not having any pigment, so there are no pigments, but yes, this does the function of storage, like storage of the food material is done by the leucoplast. So these are the different plastids that you can find in a plant cell. So components of a eukaryotic cell, so mainly the cell wall is present in the plant cell, which is made of a cellulose, and you can also find a plastid. So plastid we just now discussed. So there is a chromoplast, there is a chloroplast, and then there is a leucoplast. So these are the three types. So cell membrane is basically a lipoprotein, like you know, it is made up of a lipid, it is made up of a protein, so lipoproteinaceous cell membrane will be there. So ribosomes are, again you can find two units there. So one big unit ribosome you can find, one small unit ribosome you can find, which are having a role in the protein synthesis. Okay, then centrosome we haven't discussed yet, I'll just discuss that. Endomembrane system I discussed with you, so that is endoplasmic reticulum, Where the modification of protein is done, modification of lipid is also done. So Golgi apparatus, under Golgi apparatus, basically packaging is done. So then there is a lysosome that comes under this, where hydrolytic enzymes will be there. So they are going to act on the different old organelle, a dead and damaged cell is there, or maybe infected cell is there. So this lysosome is going to kill that particular cell. So that is by releasing the enzyme, and hence we call this lysosome to be a suicide bag. Then you can also find under this the vacuole, which is basically again arising from the Golgi apparatus itself. So vacuole is having water and other pigments. And if we are talking about the mitochondria, so mitochondria is a double membrane structure. So this is double membrane, not DM. So double membrane structure, and this is basically a powerhouse of the cell. So why do we say powerhouse? Because glycolysis is occurring there, and also the Krebs cycle is occurring in the mitochondria. Glycolysis occurs in cytoplasm. Krebs cycle occurs in mitochondria, where it produces a large amount of ATP. Okay. Then cytoskeleton is nothing but, you have seen, okay, you will study about this anyway. So there is a formation of spindle fiber while the cell division occurs. So that will contribute here, one second. So this is with respect to spindle fiber formation, which is done, one second, this is spindle fiber, yeah. So spindle fiber formation, this is basically formed during the cell division, which you will be studying in detail. So this is basically of a tubulin protein. Okay. Then some of the organelles, or mainly the organisms, if we mention. So there is a paramecium, there is euglena. So paramecium is basically having a cilia, which is again what? It is a eukaryotic cell, because paramecium belongs to the kingdom Protista. And Protista, all the organisms are what? They are eukaryotic in nature, but they are unicellular. Then flagella you can find in the euglena there. Okay. So all these are basically paramecium or the euglena. So they'll have a cilia and flagella, which is going to help them in the movement, and that is basically the movement itself. And this is basically, we can say again, this is made up of a tubulin protein itself. So protein synthesis does not occur in the mitochondria. Okay, so that way, yes. So as the mitochondria is having a ribosome, its own ribosomes are present. It is having its own DNA as well. Okay. So now inside this you can find a ribosome. Ribosome function is protein synthesis. Okay. So since the mitochondria is having a ribosome, yes, it produces a protein. Not the mitochondria alone is not synthesizing a protein there. It is the ribosome which is synthesizing a protein inside the mitochondria. Is that clear, Narendra? Then centrosome and centriole. So I'll just explain you what exactly is centrosome or a centriole. So now this is a cell, that is animal cell. Here is the nucleus. So near the nucleus you can find a centrosome which is present. So these two are centrioles which are arranged at right angles to each other. So like this. So there are many tubular proteins. So these are just line-like appearance that you can find, and that is why it is represented as a dot like this, and this is the one. And surrounding that you can find the aster rays which are arising from the centrioles. So this is the one, like this it is. Okay. So aster rays. So this basically I'll just label this. So this one is a centriole. Okay. And this one is aster rays. Okay. So these are aster rays. So both of them together, like aster ray and centriole together. So we call this to be a aster. So there is a aster formation. On the whole, this is basically a centrosome itself. So now so your name is Utkarsh. Okay. I hope you understood about this Utkarsh. Okay. So, there is a centriole and then there is a aster ray formation. So, both of them together aster. So animal cell, we call them amphiastral. What is amphiastral? Since now both the poles, now let's say these are the poles, so here is one pole and this is another pole. So you can see the aster formation, that is two of the asters are produced during the cell division process, and hence we call the animal cell to have an amphiastral condition, that is two of the asters are produced. So centrosome and centriole is basically you can find in the animal cell, and this is having a role in the cell division. Okay, nucleus, you know, this is the boss. So this is the boss of the cell, which is going to control each and every function that is occurring in the plant body or the animal body or any other organism, that is basically a living organism, where it will control the whole function of this cell. So nucleus, since it is having a DNA, so DNA is the genetic material, and this genetic material is the one that produces a protein. So that is by synthesizing, ribosomes are being coated. So basically I'll just explain you what exactly happens. So DNA is converted there into a RNA, and RNA is forming the protein. Okay, so this is basically, you will study about this in class 12 actually, but right now you can just remember DNA is the one which is coding for the RNA, and RNA is the one which is coding for a protein. So the process is called as a central dogma, and that means lot of proteins, enzymes in our body, then hormones in our body. So most of them are protein in nature. So protein coding is done by the DNA, and that is why hormonal balance is done in our body, enzymatic balance, because for different functions enzymes are required, for digestion, for respiration, for any breakdown that has to occur, so in that case enzymes are required. So for many different processes enzymes are required, and DNA is the one which is responsible for this enzyme synthesis, that is protein synthesis, by coding for RNA, and RNA is coding for the protein. So now you know the ribosomes are the ones which will be made up of a RNA, and in that case the ribosomes are the ones that are synthesizing a protein there, and hence we can say that the nucleus is the boss of the cell, because the DNA is there, and DNA is coding for the RNA, and RNA is coding for the protein. So overall control mechanism of the body, any living organism, so that is done by the DNA there. Any doubt, any of you? Any doubts that you have, you can ask. So you know about all these different organelles that are there. So these are the different organelles, and one second, let me just zoom this diagram for you all, so you can see this, right? So this is what, this is the animal cell. Okay, so in animal cell what do you see? So basically you can see nuclear envelope, nuclear pore, nucleoplasm, nucleolus is also present. So this is a cell membrane, so you can find a ribosome. So peroxisomes are also site of basically enzymes will be there. So many of the enzymes will be there, so which in turn are going to help in the respiration as well. So then you can find a cilia, slighter cilial growth you can find. So that is a hair-like structure you can see on the cell. Then there is a cytoplasm. So secretory vesicle, Golgi body, centrosome is found there, which are arranged at right angles to each other. So centriole is there. And then you can see a lysosome. Here is the mitochondria. So this one is a rough endoplasmic reticulum. This one is the smooth endoplasmic reticulum that you can find. And then you can also find, so this is a whole structure, animal cell or the cell organelles when we mention. So they have their own structure. So they have their own function. So with their own structure. So now the nuclear membrane. So that is also a membrane itself. Cell membrane. So that is also a membrane. So what exactly membrane is made up of? Membrane is made up of a protein, lipid and a protein. Okay, so fats and proteins will be there. So same way mitochondrial membrane, endoplasmic membrane, Golgi apparatus membrane, nuclear membrane. So any membrane, wherever you come across the membrane, so all these are basically made up of a lipoprotein, that is lipid, and the protein will be there. I hope this is all clear about the cell organelles. So since today's topic was basically about the cell organelles itself, so that is from eukaryotes, I discussed it. So those who anyway joined late, so you can anyway watch the recording which will be available. So any doubts that you get, so you can join me on my telegram, so that is Vani Kari ICSE class 9 and 10 biology is my telegram. Okay, so you can just search this in telegram. So this is ICSE class 9 and 10. Okay, so this you can search on the telegram and join my telegram. So any doubt you have, you can ask me there, or if you specifically need this note, you can just text me there or on the community. So I'll be sending this notes. Okay, so I hope there are no doubts. So protoplast is a cell without the nucleus, undergoing division, without the cell wall, and without a plasma membrane. So what is the answer for this? No idea? So protoplasm or a protoplast, when we mention, so this is basically cytoplasm and then the nucleus, which is covered by a nuclear, non-nuclear membrane, the plasma membrane or the cell membrane. So that means this is all a living content of the cell. So cell wall, if it is there, so this is a dead one. So cell wall is basically a dead one. So now when we mention a protoplasm or a protoplast, it is without the cell wall. So why exactly without cell wall? Because this is the dead content and this is the living content of the cell. And animal cell, directly we can call the animal cell to be a protoplast or a protoplasm, but plant cell, we need to remove this cell wall and then we can call the plant cell to have a protoplasm. So with cell wall, we don't call this to be a protoplasm. So this is directly animal cell can be called as a protoplast or a protoplasm because it is without the cell wall. So moving on to next one. Okay, so controlling center of the cell is who? So nucleus, nucleolus, mitochondria, ribosomes. Yes, that's right. So this one, so since we call this to be a boss of the cell or the controlling center, so since this is having a genetic material DNA, so DNA is in turn coding for RNA, RNA is in turn coding for protein, and proteins are the ones which are basically controlling most of the mechanism, because all the membranes are made of proteins there, all the enzymes are made of proteins, like most of the hormones are made up of protein, and hence protein is essential. So who is doing the control there? So it is the nucleus, since it is having a DNA. So moving on to next one. Yes, it is A. So nucleoplasm is continuous with the cytoplasm, that is of a cell through centriole, endoplasmic reticulum, nuclear pore, Golgi apparatus. Yes, what is the answer for this? C, yes, that's right. So this is nuclear pore. So this is a nuclear envelope. So this is outer membrane, this is inner membrane. So here is the nuclear pore. So these gaps, what you see, so these are all nuclear pore. So nucleoplasm is here inside the nucleus. Surrounding it is a cytoplasm. So cytoplasm and nucleoplasm, so they are in continuity. So that is because through the nuclear pore. So this is the answer. So moving on to next one, the main organelle involved in modification and routing of newly synthesized protein to their destination is chloroplast, mitochondria, lysosome, and endoplasmic reticulum. So where exactly modification of protein I said? So modification of protein is done. Yes, very good. So modification of protein is done in the endoplasmic reticulum. So that modified protein, let's say it can be enzyme also, which will be reaching the Golgi body, and from the Golgi body, so this is gonna reach the vesicle, so that is basically a lysosome. So this will have a hydrolytic enzymes, okay, and this is gonna perform the function of, like, killing the unwanted things or any old organelles, damaged cell or injured cell, infected cell. So all that can be killed by the suicide bag, which is basically a lysosome. So Golgi body originated from lysosome, endoplasmic reticulum, mitochondria, and cell membrane. Yes, D was the answer for the previous one. So for this one, Golgi body originated from. So from who is giving rise to a Golgi body? This is nucleus. This is endoplasmic reticulum, right? So this one is endoplasmic reticulum there. Yes, that's right. So from arising from the endoplasmic reticulum itself is the Golgi body that you can see. So this is the answer. Okay. All right. So we are done with today's session. I hope you understood everything. So any doubts you have, you can get in touch. All you need to do is you can download the Unacademy Learners app and start taking up the sessions there as well. So right now I'm conducting a quiz, but in July month we'll be having a quick discussion on different topics. So what you can do is you can download the Unacademy Learners app and start taking up free sessions which are available there as well. So now this is about the batch which is starting from 13th July. So where the mode of delivery here is English, and basically Hindi and English mix it will be. So if you are taking a subscription for class 9, so you will be getting class 10, class 11 and class 12 subscription absolutely free along with this. So you can watch the live sessions basically. So for this you need to take the subscription because these are plus classes, and we also have a offer which is for class 9, and though there are a lot of learners who start their preparation early, so that is for IIT, JEE, as well as for NEET and also for NTSE. So those students can go for this offer where so this is basically 1 year itself, and this is 34,500. With the offer this is reduced to 31,050, but with the code Vani YT, so this is the code. So with this code you will be again getting 10% discount on this particular amount. Okay, so you can go for this offer or the normal offer like the 12-month subscription or maybe 9-month subscription, that if you are planning. So for 12 months it is basically 13,800, that is with 20% discount, or otherwise the regular amount is 17,250. So if you are planning on taking the subscription for a year, that is 12 months, so with this anyway you are anyway getting class 10, class 11 and class 12 subscription absolutely free. And also since 20% discount offer is going on, so you can make a best use of this offer where it is just 13,800, where you will be saving up to 3,450. All you need to do is add the code there, that is Vani YT, to get 20% discount on the subscription. So this is about the Guardians batch which is there. So this is 1st June this started. Yes, you can watch the recordings there. So introducing community. So never miss on any update from your favorite educator. Get session notes and practice material. Share your feedback directly with the educator via poll. So you can find the link to the educator community below. So that is in the description box. So you can find the link there. So it is basically through the rough endoplasmic reticulum as well through the smooth endoplasmic reticulum in general. So that is because there is a formation. So once the modification of protein, when the modification of lipid is done in the smooth endoplasmic reticulum, so in the Golgi body, packing of the modified protein, packing of the modified lipid is done. So it is from both anyway. Okay. So check out the link to the educator community below. So this is Bucks Bounty opportunity for all learners to report any inappropriate content in the video. So be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. So thank you all. I hope you all understood. Any doubts that you have, you can ask me. And so see you all in the next session. So you can subscribe to our channel, that is Unacademy, ICSE and ISC boards. Click on bell icon to get notified about the session. Like and share the video with your friends, and download the Unacademy Learners app now. So this PDF, if you need, I can send it on the community or maybe on the Telegram, so you can join in there. Okay, so see you all in the next session. Bye bye everyone. Thank you.